energy. So the structure of phloem now, since in the previous class we have already talked about the conduction of water in the plant. We have talked about the fact how plant takes two of the pathways, apoplast or symplast. Then we talked about transpiration. We have also discussed the internal structure of leaf, in which we covered cuticle, pellicid mesophyll, spongy mesophyll, and then we talked about the entire mechanism of transpiration. So now we are going to study the other half of the topic, which is based upon the conduction of food. We have talked about the conduction of water. Now we are moving towards the conduction of food. That how a food is conducted or how it's transported within the plant. And initially, we know that it is synthesized by the mechanism of photosynthesis. We all know the mechanism of photosynthesis. We are not going to discuss it right now because it's not the part of our syllabus. But yes, so mainly the structure which is involved in the conduction of food of a plant, it is phloem. Okay, we have talked about xylem. Now the other structure which is involved in the conduction of food is phloem. So phloem, we'll study the structure of phloem in a manner. Okay, we need to look at the entire structure. We need to know, okay, how is it conducting or how is it translocating the food. So we'll cover all of it in today's class. Okay. So before looking into the process of translocation, we are doing structure. So phloem has sieve tubes. We'll see what sieve tubes are. So phloem has sieve tubes, which is made up of living cells and is accompanied by companion cell. So now when I'm talking about the structure of phloem, you need to be mindful about the structure of xylem as well. Because at times in your paper, a question is given where you have to describe the differences between xylem and phloem. Okay, so the lines that I'm underlining, these are the most important differences between xylem and phloem. That phloem has sieve tubes, number one, and xylem does not have sieve tubes. Then phloem is made up of living cells, while we know that xylem is a dead structure. Then we are also coming up with the fact that phloem has a companion cell. So there is no such companion cell in xylem. We know that xylem is a dead hollow structure, but phloem, on the other hand, is a living cell. It's made up of living cells and is accompanied by another cell as well, and it has sieve tubes. So these are primarily the three differences that you should be mindful when you're talking about the differences between xylem and phloem. Okay? Then for living cell, you need to know that it has cytoplasm, it has endoplasmic reticulum, it has mitochondria, but it does not have nucleus or ribosomes. So the living cells of phloem, they have cytoplasm, they have ER, they have mitochondria, but they do not have nucleus or ribosomes. And then the other fact is sieve elements form sieve plates, meaning the phloem has sieve plates as well. So we'll see the structure of how all of these things are made. But before looking at the diagram, we are doing the theory part. Okay? Then the sieve element has companion cells. So a sieve element has another cell which is known as companion cell, and it also has proper cellular components including cell wall, cell surface membrane, cytoplasm, vacuole, and nucleus. So the living cell of phloem did not have nucleus, but the other cell, which is accompanying the living cell of phloem, which is companion cell, it has nucleus. And it also has ribosomes and it is metabolically very active. So in these two passages, we are talking about companion cells. It's a cell which accompanies the sieve element. So sieve element does not have nucleus, while your companion cell, it has nucleus, it has ribosomes, and it is metabolically very active. So now let's look at the structure of sieve element and companion cell, because these two join in the sieve element and companion cell join to form phloem. Okay, and phloem is the structure which is responsible for the translocation of food within the plant. So I hope I'm making sense that this is when we are talking about the structure of phloem, we need to automatically jump to the fact that it has sieve element and it is accompanied by companion cells. So sieve element has sieve plates. Sometimes they ask you to label it as well. So it's very easy to identify anything which has pores in between. So it has sieve pores, right? So these sieve pores, they are the junction which enable the transport of substances. Then this structure is known as sieve plate. The gap junctions between these sieve plates is known as sieve pore. Then you can locate a mitochondria. You can locate cell surface membrane. You can locate cellulose cell wall. Internally, it is bounded with cell surface membrane. Then we can easily locate cytoplasm and ER. So we mentioned in the previous slide as well that sieve element lacks nucleus and it lacks stronoplasm and it lacks ribosomes. But since it is accompanied with another cell, which is companion cell, so basically companion cell is like a what do I say? It's also the functional unit of the phloem. The sieve element does not function alone; it functions joint with a companion cell. Okay, so these two compile or these two join together to initiate the process of translocation. Okay, so sieve element has companion cell. By the word companion, you can easily understand companion cell is there to facilitate the entire process. And so the companion cell, it has a vacuole, like you can see, it's missing in sieve element, but this companion cell has vacuole. It also has tonoplast. It has a properly arranged nucleus, which is bounded with ER, and the ER which is bounded with nucleus, it's known as rough endoplasmic reticulum. Okay? So it has rough endoplasmic reticulum. It is mitochondria. It has the important structure which now you need to know is plasmodesmata. Okay, so plasmodesmata basically allows the communication. It allows the communication between sieve element and between companion cell. Okay, so if this question can come in your P1, and this question usually comes in P2 for 1 mark, but it can come in P1 as well. Okay, anything which offers communication between sieve element and companion cell, meaning how they communicate with one another, is because of plasmodesmata. Okay, so at times I'm not sure why, but the students usually mark C4 as plasmodesmata. Be mindful that plasmodesmata is the communication between sieve element and between companion cell, while C4s are something else. It's the gap junction present between sieve plates, and sieve plate is a component of sieve element. Okay, so write it somewhere that the communication between sieve element and companion cell is held because of plasmodesmata. So the junction between sieve element and companion cell is known as plasmodesmata. Okay? This part is not plasmodesmata. This is your C4, which is the component of sieve element. I'm repeating it again and again so that you remain mindful, because this is a very repetitive mistake that I've seen multiple times. So just be mindful that sieve element and companion cell are separated by plasmodesmata and not by C4s. Okay? Then the other question which I've usually seen, it's of 3 marks. It can come for 4 marks as well, which we just discussed as well. Okay, write the differences between xylem and phloem. So the main difference is of conduction. That xylem is primarily responsible for the conduction of water and minerals, while phloem is responsible for the conduction of food. Okay, so this is the first difference. Then you can mention that xylem is a dead structure. It's not written here, but I'm telling you it's a dead structure. We have discussed this multiple times when you're doing this chapter. Xylem is a dead hollow structure, while phloem is made up of living cells. It is accompanied by companion cell, and companion cell is not present in xylem. So this could be another difference. Then the third difference could be that xylem is a hollow structure, while phloem is not a hollow structure. It's a proper cellular structure. It has all the cellular components present present inside. Then you can mention that xylem does not have sieve elements, whereas phloem has sieve elements. Then you can also mention that xylem has the deposition of lignin, while phloem does not have the deposition of lignin. right? So these could be the differences that you could easily identify between xylem and phloem. Other differences are written here, but I have mentioned the important ones, the most essential ones that should be there in your script. And you can also mention that this is a one-way only transport, while in phloem, it's a two-way flow, meaning the food is just not, if the food is made in leaves, then food has to be transported towards the stem as well, because stem is a living component of the plant. Okay, so it's a two-way flow. Okay, if it is needed in leaf, then it's also needed in stems. It's also needed in roots. So it's a two-way flow. While water is just conducted towards the upside, towards the leaves, and towards all the structures that are present above. Okay. Now moving towards the translocation. So this question is there in your paper for 5 marks. Okay. So everything that should be a part of your script is written in my slides, and make sure that you write all of the things that are written here, because this is a compilation from your past papers, from the marking scheme of your past papers. So this is what your examiners are expecting you to write, and this is present in my slides as well. Okay? So if you understand this, you write this, you get 5 on 5. And it's a very easy question because you do not have to use your brain to write something else. Everything is in the slides, just study them and write it in your paper. Okay? So translocation is a process where we are now going to talk about the transport or the conduction of food. So after photosynthesis, meaning कि photosynthesis process के बाद your glucose is synthesized, right? The glucose for plant is synthesized. And after photosynthesis, when sugars are formed, they are transported from mesophyll to phloem. So in step number 1, we mentioned this in the previous class. In mesophills, it's the main site of photosynthesis, because in palisade mesophills, there's a huge quantity of chloroplast, right? So palisade mesophyll is the main site of photosynthesis. There, the process of photosynthesis has taken place. Then the sugar is formed, and it's now transported from mesophyll to phloem. We just discussed the structure of phloem. Okay, so this is that structure which is responsible for translocation. Now, then they may move by symplast or apoplast. So it's also a passage that we have mentioned in the previous class as well. The symplast is the mechanism in which the substances move inside the cell through the cytoplasm, while in apoplast the substances move from cell wall to cell wall, right? Then the companion cell and sieve element work together, because sieve element cannot function alone. It needs companion cell. Then the sucrose is loaded in companion cell or in sieve element by active transport. Okay, so since companion cell and sieve element, they are the components of phloem. Now we are saying that from mesophyll, sucrose is loaded either in companion cell or in sieve element by the transport mechanism of active transport, meaning that this process requires energy. Then this is the most important uh, this is the most important line that should be in your answer script, because if this is not written, then you automatically you'll lose 2 marks. Okay, so make 2 3 stars for this point, write it somewhere else, write it in your notebooks, or I don't know, wherever you are taking your notes, that this should be there when you're talking about translocation. It says that hydrogen ions are pumped out of companion cells into their cell walls, which creates excess of hydrogen ions in the extracellular compartment. So extracellular compartment is the region which is outside the cell. When the process of loading sucrose inside companion cell or sieve element has taken place, hydrogen ions are present in your companion cell. Okay? Once sucrose is loaded, hydrogen ions are pumped out from the companion cell. They're pumped out from the intracellular compartment to the extracellular compartment, and that extracellular region is cell wall of the companion cell. So it has been done for some phenomena that needs to take place. It's done so that this concentration gradient could be built up. And when hydrogen ions, now that they're pumped out from the cell to the cell wall, it has created the excess of hydrogen ions in the extracellular compartment. And then sucrose is transported by port transporter into companion cells against the concentration gradient. So why there's this reason, okay, why hydrogen ions have been pumped out. They have been pumped out to create concentration gradient. वैसे तो they're not going to ask you this, कि why it has been pumped out, because it's a mechanism, and but the reasoning for why they're pumped out is to create concentration gradient. So now when sucrose is transported by co-transporter, it's entering into companion cells. In, if you want to say this in simpler words, the sucrose is transported with the process of active transport by the help of co-transporter into companion cells against the concentration gradient, because we are mentioning active transport. So for active transport, we need to move substances against the concentration gradient. We use the word down the concentration gradient for diffusion, right? But since we have not involved energy, we need energy. We are using the process of active transport. We'll talk about the concentration gradient going against, right? Then sucrose then moves from companion cell into sieve tubes through plasmodesmata. Okay, if sucrose has been loaded into companion cells, it will move to sieve tubes through plasmodesmata, which I just mentioned in the previous slide as well. Okay, what is the usage of plasmodesmata? It basically offers communication between two of them. Then in the source, so I'm sure this is a new terminology for you. In the source, sucrose is loaded into phloem, resulting in decrease of water potential. I'll explain what source is, so do not worry about it. Just understand what I'm saying. In the source, sucrose is loaded into phloem, resulting in decrease of water potential. This is something very important. As the sucrose is entering into your sieve element, the water potential of the sieve element will decrease, right? Because as we'll add solute, the content of the solvent content will reduce, right? So sucrose is loaded into phloem, resulting in decrease of water potential. So this water potential has been created for a reason. Then the water will enter the sieve elements, moving down water potential by osmosis. Okay, like for example, if this is my phloem, I have loaded sucrose in this. Okay, I've added food content in this. So automatically the water content present inside this phloem will decrease. And then we know that the process of osmosis is continuously taking place. जैसे ही water content reduce in this phloem, water will move through the process of osmosis down the water potential gradient. So water is going to add into this, and then it has been added to create pressure difference. So pressure difference is created between source and sink, and then it will cause a mass flow of water and solutes. By solutes, I mean sucrose. So it will cause a mass flow of water and solutes from high to low pressure, and at sink, sucrose is used. So sink is basically the region where food is needed, and source is basically the region where the food has been made. Okay? Source is the region where food has been made, yani where sucrose has been made, it's the source. And for example, if sucrose is needed at roots, then roots are going to be sink. Okay? So source and sink are two terminologies which define where food has been made and where food has been utilized. So now I'm pausing, and if you have any question, you can unmute yourself.